Greetings to the viewers of this video. In this video, we are going to see the topic of rate-controlled drug delivery systems. Specifically, we are going to see the rate pre-programmed drug delivery systems. First, let us know why we need new drug delivery systems. We know that developing a new drug takes off lots of time and money. So, developing a new drug delivery system is preferred. Due to the difficulty in developing new drugs, more and more emphasis has been given to developing new drug delivery systems for existing drugs as well as new chemical entities. There are three modes of drug delivery: targeted delivery, controlled release, and modified release. Now let's see why we need to control the drug delivery system. It has been introduced to overcome the drawback of fluctuating drug levels associated with conventional dosage forms. The controlled release of a drug involves releasing the drug at a predetermined rate at a specific interval and having specific release profiles. The other name for controlled release systems includes smart, targeted, intelligent, novel, and therapeutic. A controlled release system usually operates on three things, namely, advanced engineering principles, a control approach, and consisting of logic elements with or without a sensor. Before going into the topic of rate pre-programmed delivery system, we must know something about the diffusion process, as this rate pre-programmed drug delivery system works based on Fick's law of diffusion. Let's now see the diffusion theory. Diffusion can be defined as a process by which molecules transfer spontaneously from one region to another in such a way as to equalize chemical potential. Here you can see the tablet contents get diffused into the solution without any external energy. The migrating molecules are called diffusants. They can also be termed as permeants or penetrants. The membrane or matrix in which the diffusant migrates is the diffusional barrier. The external phase is called the medium. The concentration gradient of the diffusant within the diffusional barrier is the driving force for diffusion. Now, let us see Fick's first law of diffusion. It states that movement of particles from high to low concentration is directly proportional to the concentration gradient. J is directly proportional to dC divided by dx. Removing the proportionality, we introduce the alphabet D to the formula. The negative sign indicates that the flow is from high concentration to low concentration. Fick's first law is mostly applicable in the case of steady-state diffusion, that is the concentration of atoms passing through remains constant, it does not change with time, as such in zero-order release of drugs. J is diffusion flux, C is concentration, D is diffusion coefficient, and dC by dx is the concentration gradient of the particle. Fick's second law is applicable to non-steady state diffusion. It states that the change in concentration with time at some point on a concentration gradient depends on the second derivative of the concentration gradient. The law goes like this: dC by dt is equal to d multiplied by d square c by dx squared. dC is a change in the concentration of the particle. dt is a change in time. dx is a change in position. d is diffusion coefficient. dC by dt is a change in concentration with time. d square c by dx squared is the second derivative of dC by dt. Based on technical sophistication, rate-controlled drug delivery system is classified into rate pre-programmed, activation modulated, feedback regulated, and site targeting. The rate pre-programmed drug delivery system is classified into polymer membrane permeation-controlled, polymer matrix diffusion-controlled, and micro-reservoir partition-controlled drug delivery system. For the rate-programmed delivery system, we are going to see only the three basic things. How is the drug reservoir fabricated? How is the rate of drug release calculated? What factors influence pre-programmed drug delivery? Now, let us see about polymer membrane permeation-controlled drug delivery systems. A drug formulation is totally or partially encapsulated within a drug reservoir compartment. The drug release surface is covered by a rate-controlling polymeric membrane having a specific permeability. The drug reservoir may exist in solid, suspension, or solution form. The polymeric membrane can be fabricated from a non-porous or microporous membrane. The encapsulation of drugs inside the reservoir compartment can be done by injection molding, spray coating, microencapsulation, or other techniques. The release of drug molecules from this type of drug delivery system is controlled at a pre-programmed rate by controlling the partition coefficient and diffusivity of the drug molecule and the thickness of the rate-controlling membrane. The rate of drug release is calculated by this formula. K is the partition coefficient, D is the diffusion coefficient, Cr is the drug concentration in the reservoir compartment. There are three types of polymer membrane permeation-controlled drug delivery systems. They are sphere type, cylinder type, and sheet type. The cylinder type is made with a non-porous membrane, and the sheet type is made from a porous membrane. These are the representatives of the polymer membrane permeation-controlled drug delivery system. Progestasert IUD is an example of cylinder type. Progestasert system is an example of sphere type. Norplant subdermal implant, transderm nitro are sheet types. In the Progestasert IUD, the non-porous membrane is made up of ethylene vinyl acetate copolymer and the drug reservoir contains 38 mg of progesterone microcrystals suspended in silicone oil. It delivers progesterone in the uterine cavity at a rate of 65 micrograms per day, which gives contraception for 1 year. Next, the polymer matrix diffusion-controlled drug delivery system. The matrix system consists of active and inactive ingredients mixed homogeneously in a dosage form. They can be divided into a non-swellable matrix and a swellable matrix. This swellable matrix type tablet is prepared by mixing drug and water-soluble hydrophilic polymers such as HPMC, sodium alginate, and other excipients. This is a schematic representation of the swelled hydrophilic polymer matrix. This is a glassy phase of the matrix. This is the gel phase of the matrix. And we have diffusion, swelling, and erosion front in it. Then, non-swellable matrix. Water-insoluble hydrophobic polymers, such as polyvinyl acetate, is mixed with drugs and other excipients and compressed into a tablet. The matrix does not swell, but there forms a drug depletion zone that releases the drug. The matrix is insoluble and shows no disintegration and no swelling. Now, let us see the transderm nitro. In a cross-sectional view, this is the drug reservoir. It is prepared by homogeneously dispersing the drug in the rate-controlling polymer matrix. This is the drug impermeable metallic plastic laminate, rate-controlling polymeric membrane, and adhesive layer. If the rate-controlling polymer is lipophilic, then it is non-swellable matrix. If it is hydrophilic, it is a swellable matrix. The drug dispersion can be done in the following three ways. Finely ground drug particles are blended with liquid polymer. Mixing of the drug with rubbery polymer at elevated temperatures. Dissolving the drug and polymer in a common solvent, followed by evaporation at elevated temperature or under vacuum. The rate of drug release is calculated by this formula. A is the loading dose in the polymer matrix. Cr is the drug solubility in the polymer. dP is the diffusivity of drug molecules in the polymer matrix. The release of drug molecules is controlled by controlling the loading dose, polymer solubility of the drug, and drug diffusivity in the polymer matrix. Advantages of the matrix system. It is very easy to fabricate in wide range of size and shape. The matrix system is suitable for both degradable and non-degradable systems. No danger of dose dumping in case of a rupture in the non-swellable matrix. Disadvantages of the matrix system. The achievement of zero order with this type of delivery system is difficult. Not all drugs can be blended with a given polymeric matrix. Water-soluble drugs tend to burst from the system. Next is the micro-reservoir partition-controlled drug delivery system. Simply it is a combination of matrix and membrane type system. The reservoir system consists of a core and a coating membrane. The core contains active ingredients and excipients, and the membrane is primarily made up of rate-controlling polymer. An aqueous suspension of the drug is microdispersed by a high-energy dispersion technique in a biocompatible polymer to form homogeneous dispersion of many discrete, unleachable microscopic drug reservoirs. Next is coating of the drug. Depending on the desired rate of release, it can be further coated with a layer of biocompatible polymer to modify the mechanism of drug release. The rate of drug release is given by this formula. Here K is the partition coefficient, D is the diffusivity of the drug, H is the polymer coating membrane thickness, S is the solubility of the drug. This is the cross-sectional view of deponent transdermal patch containing glyceryl trinitrate. This has a drug impermeable laminate and the drug reservoir gradient. Adhesive layers of different thickness, which releases the drugs at the desired rate. This patch contains 18.7 milligrams of glyceryl nitrate and the average amount of drug absorbed in 24 hours is 5 milligrams. The key variables for reservoir systems include levels of polymer, levels of pore former in film coat, drug load, and solubility. The advantages of a micro-reservoir system include: achievement of zero-order release is easy. Very easy to fabricate in a wide range of sizes and shapes. And drug inactivation by contact with the polymeric matrix can be avoided. The main disadvantages are the dangerous dose dumping. The solution behavior of reservoir system-based products may change on storage. This may be minimized by adding a curing step at the end of the coating process. Curing is a chemical process employed in polymer chemistry and process engineering that produces the toughening or hardening of a polymer material by cross-linking polymer chains. Thanks for watching. Have a great day.